Hello everyone. In this lab, we will learn how to determine the dissolved oxygen DO using both the Winkler titration and DO meter measurement. We will also study the effect of temperature on DO saturation levels in water. Dissolved oxygen refers to the level of free, non-compound oxygen present in water. It enters water through the air or from aquatic plants as a result of photosynthesis. Aquatic organisms cannot use oxygen in water molecules H2O to breathe. They instead rely on free molecules of oxygen gas O2 dissolved in water. For the dissolved oxygen determination, we have two methods, the chemical method, Winkler titration, and DO meter measurement. In this lab, we will use both Winkler titration and DO meter to test the water samples prepared at various temperatures, 35 degrees Celsius, 50 degrees Celsius, and room temperature. Please wear all required PPE. This includes safety goggles, a lab coat, gloves, and tight long hair. Note that glass bottles can cut fingers, so always hold them in the middle. Be careful of water on the floor in this lab. Walk slowly. In the Winkler titration method, the oxygen concentration is determined through a series of chemical reactions. First, oxygen fixed. Second, produced iodine. And third, titrating the I2 using a sodium thiosulfate solution. Based on the concentration and volume used of sodium thiosulfate, the oxygen concentration can be calculated. Using a disposable plastic transfer pipette, add 2 milliliters of MnSO4, a pink solution. Also note that the disposable plastic transfer pipette has a capacity of 1 milliliter. Using a fresh disposable plastic transfer pipette, add 2 milliliters of alkali iodide azide reagent. Insert the glass stopper and drain off any excess liquid. Mix by inverting several times. A brown precipitate, MnO2, should form if dissolved oxygen was present. Allow the bulk of the precipitate to settle into the bottom half of the DO bottle. This should take about 5 minutes. Remove the glass stopper and add 2 milliliters of concentrated sulfuric acid to the sample. Insert the glass stopper and drain off any excess liquid. Mix by inverting several times to dissolve the precipitate. Transfer the solution into a 204 milliliter volumetric flask. Fill the flask to the mark. Transfer the contents of the 204 milliliter volumetric flask into a 500 milliliter Erlenmeyer flask. Write down the initial volume reading of titrant and start titration until the color of the sample has turned light straw yellow. Add 5 drops of starch indicator. Addition of the starch indicator should cause the solution to turn blue or black. Continue titration until the colorless endpoint is reached. Write the final volume reading. Based on the volume and concentration of titrant, you can calculate the concentration of oxygen in the water sample. In the previous section, we talked about determination of DO using titration method. Now, we will teach you how to determine DO using a DO meter. Following is the operation procedure. First, fill a BOD bottle with air-saturated room temperature water. This should be done slowly and make sure there are no bubbles in the bottle. Once the water reaches the middle of the bottleneck, stop filling and put on a stopper. This is your room temperature sample. Put the bottle in the holder, which is attached with the DO meter. Rinse the bottom membrane part of the DO meter with deionized or DI water. Open the sample bottle stopper and put in the DO sensor quickly. Turn on the stir by switching the red button. Wait about 5 seconds and read the number. The DO reading may not be stable, so just take the reading at 5 seconds. Finally, stop the stir, take out the DO sensor and rinse it, and put it back in the DO sensor storage bottle. Please note the DO meter was calibrated by the technician before you came to the lab. In real work, you need to calibrate the meter before using it. In today's lab, you will prepare 2 room temperature samples. The technician has already prepared 2 samples at 35 degrees Celsius and 50 degrees Celsius. You need to determine the DO using both the titration method and DO meter. Using the following equation, you can calculate the expected DO concentration for each aeration temperature and at ambient atmospheric pressure. Compare them and calculate the error. After this lab, you should know how to determine the DO using both the titration method and DO meter. Join us next time for the 5th and final lab, chemical oxygen demand and total organic carbon.